Well, good morning, AP Bio students. Mrs. Shelton here, and just going to give you a quick Screencastify for your use on Chapter 4, A Tour of the Cell. As you guys probably know at this point, there's a lot of content in here that you are familiar with from 7th grade life science class and also from your freshman year of biology. But these are the you must knows that I've covered on your notes as far as what we really need to be taking out of this chapter and making sure that we master at this point in AP Bio. So making sure we understand differences between prokaryote and eukaryote cells, also just knowing the structure and function of organelles that are common in plants and animals, but then what's different in plants and animals also is important as well. But then also how form fits function when it comes to a cell, how your surface area versus volume actually affects then rate of nutrient intake, and how each cell is going to differ in what types of organelles and subcellular components it actually has. So these are the things we need to be able to take out of this chapter and be able to apply. So yes, first you need to make sure that you're knowing the organelles and what they do and be able to recognize them, but can we apply that to our concept of form fits function and how surface area to volume ratios do affect cell efficiency in order to answer that ultimate question of why are cells small? So let's go through the notes here. Let's go ahead and turn these, get the presentation going here so we can see this really cool picture, in my opinion, because I think this is awesome as we are looking at, come on, we got it. There we go. So what we're looking at here is actually a neuron. So this is a motor neuron that is innervating with skeletal muscle fibers. And you can definitely see that they're unique in their shape because for a neuron, form has to fit function. And so here we're actually seeing the neuromuscular junction in this in order to be able to control that muscle fiber contraction. Now, this is with a scanning electron microscope. What we can't see in here is we can't see the acetylcholine that's being released and the receptors that are here. Biology is so cool. I'm a nerd, but it's pretty awesome, just the way that things work. But we'll start to see it on the cellular level. Now, we do have to remember that cells are small. And they are small for a reason in order to be more efficient. And what we do need to make sure that we are understanding is relative size, and a light microscope is going to allow us to be able to see most eukaryote cells. Now, bacteria, on the other hand, bacteria are fairly small in comparison, so we're not going to be able to see a lot of detail when it comes to looking actually at organelles that are much smaller or even looking at prokaryotes. So we can see that they exist, but for details, that's when we need to make sure that we're now using an electron microscope. Electron microscopes are going to help us. So just know relative size. You are never going to be asked to name the number, but we just need to know size-wise in comparison to everything else. Now, here are just some cool pictures with light microscopes. Recognize that there's lots of different staining techniques that can be used with light microscopes. These would be typically the types of the staining that we would do with light microscopes in our lab. And electron microscope pictures we can see down here, where now we're getting up close and we're even seeing like cilia on a cell that's used to actually move materials past the cell. This would be the transmission electron microscope, where you can now actually see internal structures, where your scanning electron microscope is going to give you the surface pictures. Now, one of the main concepts that we need to make sure that we are mastering here is we need to make sure that we are understanding this unity of all life. And in order to understand the unity of all life, we need to take a look at our basic prokaryotes and recognize from prokaryotes what every single prokaryote has to have in it, and then the fact that every cell on this planet has to have that too. So when we take a look, a prokaryote, it's a cell, so it has a plasma membrane. So every cell has a plasma membrane. Some have cell walls, some don't, but every cell has a plasma membrane around it, the cell membrane. Every cell has to have DNA. So in here, though, in a prokaryote, DNA is not housed in a nucleus, so there's a difference there. It's housed in a nucleoid region. And also you have to know that the DNA, or the bacterial chromosome, is circular, which is really important too. And if you have DNA, well, then you got to have ribosomes for turning that DNA into the protein. Now, we're also going to have cytosol and enzymes for respiration, for DNA transcription and translation, when we talk about DNA replication, and that's all going to be here in the cytosol. Some cells are going to have flagella, some aren't, but the commonalities that every cell is going to have is plasma membrane, DNA, ribosomes. Every cell is going to have that. Now, when it comes to a prokaryote, though, recognize too, they're very small. Because they don't have membrane-bound organelles, they are not as efficient at communicating with other cells and have that ability. So we're going to have single-celled organisms, but they also are very efficient at replicating really fast because they're very, very simple. Now, when we do talk about the plasma membrane, chapter 5, we're going to go into a lot more detail of the plasma membrane, but your plasma membrane is composed of a lipid bilayer of phospholipids, where we have a hydrophilic region of these heads that have that phosphate group on them, and then the hydrophobic region of these fatty acid tails. And then in chapter 5, we'll talk more about these integral proteins and what they do as part of the plasma membrane. With cells, one of the skills you guys are going to have to be able to do is calculate your surface area to volume ratios and understand too that if I have a cell that needs to grow and the organism needs to grow, say by 5 times, it is much more efficient to actually just increase cell number instead of just making one cell bigger. And the reason for that is when we look at surface area is the equivalent of the plasma membrane. The job of the plasma membrane is to regulate the nutrients and waste that go in and out of the cell. The volume is what we're talking about, the cytosol, all of the organelles needs, all of the metabolic needs that are inside the cell. We need to recognize that as something gets larger, surface area and volume do not increase at the same rate. And that's just basic math. Volume is calculated on a cubed basis. I'm going to multiply my height times width times length. My value here would be in a unit cubed is what I would have for volume. Surface area, I'm just multiplying 2 dimensions together and then adding all 6 sides. And so my value here would be in a squared value. So do recognize, like when I do look at this box, if this was, just boxes are easier to calculate, my surface area to volume here is a 1.2 surface area compared to volume, where now if I increase just number of cells, I can maintain way more surface area compared to volume, so the cell can be more efficient at getting nutrients and releasing waste. Here's another example of calculations. In our osmosis diffusion lab, we will do a few more calculations of calculating surface area to volume and making sure we're seeing that surface area to volume ratio as a cell gets bigger. I mean, here at 1 centimeter, a 1 centimeter by 1 centimeter by 1 centimeter cube, my surface area to volume ratio is a 6:1 ratio. So now, if I go up to a 2 by 2 by 2, well, now my surface area to volume ratio is a 3:1. I have less surface area compared to my volume. Again, I'm using volume as my denominator here so I can make sure that I am truly looking at how much surface area there is per inner cytosol and cytoplasm. Here I go by a 3 by 3 by 3, and now my surface area to volume ratio is a 2:1. So you're seeing that we are losing surface area compared to volume as we increase in size. Now, if we take a look at a eukaryote cells are much bigger. They have their DNA enclosed in a nucleus. They have membrane-bound organelles. The DNA of a eukaryote cell is rod-shaped instead of circular like in prokaryotes, but there's still DNA. There is still plasma membrane. There's still ribosomes like every other cell, but now we actually have specialized structures inside that allow for more cell specialization and more efficiency in performing different jobs and tasks. Enzymes can be centralized in order to, again, make for that efficiency. So when we do take a look at a eukaryote cell, this is an idealized animal cell. You are never going to see this cell in the real world. It's just idealized to show you every possible combination of organelle that can exist in a eukaryote cell. When we look at a plant cell, plant cells are also eukaryote cells too. But what I do need you guys to recognize with a plant cell is a few differences in a plant cell. Plants have additional components that an animal cell does. A plant cell is going to have a large central vacuole, and that large central vacuole is going to be where water and nutrients are stored. It also allows for increased turgor pressure against the cell wall. That's another difference. Is outside the plasma membrane is the cell wall composed of cellulose. And then also plant cells are going to have chloroplasts in order to perform photosynthesis. Again, I told you guys in lecture, but I want to make sure that you're crystal clear. Plants have both mitochondria and chloroplasts. Do not think that it's just animals that have mitochondria and plants just have chloroplasts. Plants have both. We need plants. Plants don't need us for energy. Now, in looking at a cell, a eukaryote cell, the nucleus is incredibly important in order to differentiate a eukaryote versus a prokaryote. And inside my nucleus is where my DNA is. And during interphase, the normal phase of the cell, DNA is arranged in chromatin. Now, to us, it looks like unorganized DNA because it's not now all bound up into chromosomes that we can see. We're only going to see chromosomes and DNA in chromosome form right at cell division. But during the normal phase of the cell, it's going to be in this chromatin structure. It's still highly organized, but under a microscope, we're not going to be able to see that. Now, inside the nucleus also, there is a more dense region that's called the nucleolus. Now, why this is more dense is because this is a region of a high amount of transcription and making RNA. So RNA, and especially ribosomes, because ribosomes are RNA and protein complexes, these are made here in the nucleolus. Now, the nucleus has a membrane structure around it. It's called the nuclear envelope. And then there's also these nuclear pores, so ribosomes can get out, messenger RNA can get out, but DNA always stays inside the nucleus. Now, what we're also seeing too, attached to the nucleus is rough ER. So this rough ER is really important and a really important part of the endomembranous system. So I'm going to have ribosomes produced here in my nucleus, but then ribosomes then come out and they'll either attach themselves to the rough ER, we call them bound ribosomes then, and this rough ER with those bound ribosomes, this is going to be the site of a high level of protein synthesis. So translation is going to be taking place here, and we're going to get that primary structure of our proteins occurring here in this rough ER. So if I have these bound ribosomes mean they are embedded or attached to the membrane of the rough ER, but also ribosomes can be free ribosomes too, and free ribosomes would then be loose out in the cytosol. These would be ribosomes that would be for making proteins that are destined to stay inside the cell and be used in the cell. However, a lot of the rough ER, the proteins made here, are going to then get produced and modified and perhaps ultimately shipped out of the cell. So if we take a look, this is more up close. So here's my nucleus, here's my nuclear envelope, my nuclear pores, and this is attached then directly to the rough ER that has all of these bound ribosomes on it. When we do take a look at a ribosome, the ribosome structure kind of looks like this, this double cookie here, where I've got a great big cookie on top and I've got a small cookie here. It's actually in two subunits, but right now what I need you guys to recognize is that it is an RNA structure with proteins as well. So it ends up serving as an enzyme for, during protein synthesis, to perform translation. Now, what I'm also seeing in this particular picture, so here is my rough endoplasmic reticulum. Now, my rough endoplasmic reticulum is going to be the site of protein synthesis with the bound ribosomes. Now, primary structure is going to be formed here, possibly secondary, possibly some tertiary structure. But in order to get modified fully, what has to work together is the rough ER with the Golgi apparatus. And proteins need to get packaged and shipped to the Golgi apparatus. So what I'm seeing coming off here is a transport vesicle. So proteins get wrapped up in a transport vesicle, which is essentially, this is just an exocytosis where the membrane that made this up is wrapping around the proteins in order to now transport it to the Golgi apparatus. So the rough ER has this one job of protein synthesis. Now your smooth ER, on the other hand, is not about protein synthesis. This is where a lot of your cell's metabolic activities occur and detoxification occurs. So when we're processing carbohydrates, when we're processing lipids, these are going to happen here in the smooth ER, and detoxification is going to be happening with the smooth ER too. So a cell like in the liver, and what I just actually named was like the function of the liver, well, your liver cells are going to have a very, very high amount of smooth ER because that's what they need to be doing, which is that lipid metabolism, that carbohydrate metabolism, and that detoxification. Where now, if I have a cell that makes a lot of proteins, so like some of your glands that are producing lots of hormones that are proteins, I'm going to have a very high concentration of rough ER is what I'm going to have in cells like that. Now, like I said, with proteins that are initially formed in the rough ER, it's through transport vesicles that they are now shipped to the Golgi apparatus. And membranes are fluid, and we'll learn more about that in chapter 5, but these membranes then fuse with the Golgi apparatus, and that's what the Golgi apparatus is too, is these series of membranes that have then lots of enzymes in here that can then modify these proteins for whatever function they need. So a lot of the proteins that are being formed here are destined for the cell membrane to be a transport protein through that plasma membrane or for export of the cell as well. But the Golgi apparatus then is going to modify those proteins and then actually ship those proteins to the cell membrane again through transport vesicles. Now, sometimes what ends up happening is there's enzymes that are digestive enzymes. Now, your textbook would describe them as hydrolytic enzymes, enzymes that can perform hydrolysis. Now, these digestive enzymes bud off of this endomembranous system from the nucleus to the rough ER to the Golgi apparatus, but they are packaged then in plasma membrane. So they're not just loose and digesting everything in the cell. That would be bad. But these lysosomes, we do need to recognize these are these little packets of digestive enzymes that are out here in the cytosol, and their job is to digest material. So here's an example of a cell that is engulfing a food particle, pulling it in, and then the membranes can fuse together in order to then break down those large macromolecules. This, if we talk about something like lysosomes, some of your macrophages, your white blood cells that are just gobbling up old dead cells or foreign pathogens, they are packed full of lysosomes in order to then do endocytosis, pull those particles in, but then digest them and break them down. So here's just another example then of lysosomes fusing with an organelle or another organism in order to then digest it. Now, in a plant, so here we're seeing this large central vacuole. Now, animal cells do have vacuoles too, and a vacuole is going to look just like a lysosome under a microscope. It's the contents that then becomes different in an animal cell. Your vacuole is going to be little storage packets of nutrients or water or waste or things like that, whereas a lysosome is going to have hydrolytic enzymes, digestive enzymes. So in an animal cell, a lysosome versus a vacuole is going to look the same, and you are not expected to understand that difference just in function. But in a plant cell, you should definitely tell the difference here in the large central vacuole. So it's this giant swimming pool in the middle, and it actually has its own membrane called a tonoplast that surrounds that vacuole that has its own transport, its own waste and nutrients in here. And this is really essential for actually controlling cell structure and making sure that the cell maintains rigidity. So here I'm seeing my endomembranous system here, this continued nucleus where transcription is going to happen. I've got my rough ER where translation is going to happen, and then I'm going to have my protein modification that starts In the rough ER, I'm going to have transport vesicles that actually then carry those proteins to the Golgi apparatus, and the Golgi apparatus is going to then modify those proteins either for release as lysosomes in the cell or fuse with the plasma membrane for export of the cell. But here's my transport vesicles is another term that we should make sure that we're understanding. Your smooth ER, I'm going to recognize my smooth ER because one, it's smaller membranous tubules, but I also don't have any ribosomes attached to it. Now, the next concept that we need to make sure that we are understanding is that we're understanding the endosymbiont theory of development of eukaryote cells. So, and really what this is addressing is the fact that eukaryote cells that have mitochondria or mitochondria and chloroplasts, how they obtained these organelles is through this endocytosis of a cell engulfing another prokaryote cell that actually had this ability to perform aerobic respiration or performed another prokaryote that had the ability to perform photosynthesis. Now, our evidence of this, because remember that that's the job of the mitochondria. Mitochondria is the site of aerobic respiration. We'll cover lots of details about it. Chloroplast is your site of photosynthesis. But our evidence of endosymbiont theory actually exists just by looking at typical mitochondrial structure. Mitochondria is a double membrane structure. And this double membrane structure can be achieved through that endocytosis. But the other piece of evidence that we do have is the circular DNA and the ribosomes inside this inner membrane that we find inside of the mitochondria. Now remember, circular DNA was found in prokaryotes. So in this inner structure, I have plasma membrane, I have circular DNA, and I have ribosomes. And so that exists in all of our eukaryote cells that can perform aerobic respiration. A chloroplast too gives us evidence of this endosymbiont theory, where again, double membrane structure that's achieved through this endocytosis action. The double membrane here does not actually even serve a purpose. It just has a double membrane, where the double membrane in the mitochondria does serve a function, does serve a purpose. But now inside, I have the stroma, which is kind of like a cytosol solution of enzymes. But I also have circular DNA and I have ribosomes, again, in my chloroplasts that perform photosynthesis. So understand that endosymbiont theory and the evidence that both a mitochondria and a chloroplast do give us for that endosymbiont theory. Now, I do have one other structure that is necessary for you to know, and this is a peroxisome. Now, peroxisomes are actually going to be found, these are these membrane enclosed series of enzymes that are going to be found next to mitochondria or next to chloroplasts. And what the peroxisome does, now you won't have to recognize it in structure because it actually just looks like a little vacuole or it looks like a little lysosome, but this contains enzymes that are responsible for breaking down peroxide. Now, why they're next to the mitochondria and chloroplast is we use a lot of free protons. A lot of free protons get used, and we'll talk about that again when we talk about respiration and photosynthesis. But some of the excess protons end up forming peroxide with water. And so peroxisomes are essential for breaking that peroxide down into water and oxygen and making sure we're detoxifying the cell. So that's its job. Now, the next section that you're going to read in the book on 4.6 is really talking about the cytoskeleton. And when we do talk about the cytoskeleton, cyto meaning cell, skeleton meaning internal structure, we should just be aware of the network of proteins and fibers and tubules that exist in order to maintain shape or to even have some motor action. So, and here's just kind of a cool picture here of motor proteins that are moving across this microtubule of the cytoskeleton, kind of like a road, like, oh, we need to get to another location. And so the events that actually happen inside of a cell are pretty cool. But I just need you in general to recognize when I'm talking about cytoskeleton, I'm talking about fibers or tubules that are all proteins that help maintain internal structure. And they're different based on their size. So here's just an example of a structure that I'm sure you're familiar with. In animal cells, there are centrioles. And these centrioles enable a cell. We're going to see centrioles. We're going to see these centrioles separate during cell division. And these are composed of these protein microtubules, is what they are. So there's these two structures that exist, but then right at the beginning of mitosis or meiosis in an animal cell, they're going to move to opposite sides of the cell. But those are formed with microtubules. If we talk about cilia or flagella and flagella that need to move. So the difference between cilia versus flagella. Flagella, usually there's just one long single flagella that's for motility. Cilia are usually very numerous, but these are extensions then from the cell that actually move in order to create movement. Now, when we take a cross section of any type of cilia or flagella, even if we looked at a prokaryote versus a eukaryote, they're all going to have this 9+2 arrangement. And this 9+2 arrangement of these motor microtubules, these motor proteins, is another piece of evidence of evolution that all of our flagella contain this 9+2 arrangement. Now, I can have additional folds that do exist in the cell membrane and the plasma membrane. This is where I get villi and microvilli to increase surface area. Because again, surface area, the more surface area I have, the more efficient that particular cell is at taking in nutrients or getting rid of waste. So cells that actually line your small intestine are going to have lots of extra folds and additional folds in them in order to be more efficient at absorbing the nutrients that you are taking in. And in order to help maintain that structure, we have these little microfilaments. So microfilaments are just these smaller fibers of the cytoskeleton to help maintain that structure. Now, in 4.7, we do need to be familiar with what is referred to as the extracellular matrix. The ECM is what you should be familiar with. Now, here is my cell. Here's my plasma membrane with a lipid bilayer. Here I'm getting another example of cytoskeleton. Here's some microfilaments that are helping to maintain the cell shape. But inside the cell versus outside the cell, there's a lot of proteins that are produced inside the cell and then pushed out to the outside to make up the extracellular matrix. So in cells like your bones, most of your bone is actually extracellular matrix, that you had these little osteocytes that were just producing these collagen fibers and producing then the mineral components and assembling those and pushing it out of the cell in order to give that rigidity of your bones. But this is extracellular matrix outside the cell. These are just examples of different types of protein that we could see, plus other components that were assembled in the cell and then pushed out for different purposes. Most of your connective tissue in your body, connective tissues in your body have lots of extracellular matrix of different types and varying types, but that's outside the cell. Your cell wall is actually a part of extracellular matrix. So cell walls, not in us, in plants, are, remember, made of that cellulose. So that polysaccharide, that's that structural polysaccharide. Now, what we also see in the cell wall, because the cell wall provides this structure and support for a plant cell, but we also want to make sure that nutrients can easily pass from cell to cell. So what we actually see in plant cells is there's these little tubes that have been built in, or these little holes that have been built into the cell wall in order to allow for nutrients to pass. These are called plasmodesmata. So it's important that you do know that term, this plasmodesmata. This is plant cells only, but in plant cells, there are these holes, these little tunnels that go through cell walls, so this cell can share fluids and nutrients with this cell, etc. So here's my picture of my extracellular matrix. Here's my lipid bilayer. And then the last thing we need to talk about is now in other cells, so not in plant cells, just understanding that cells in multicellular organisms need to be able to either be really tightly compact. So we've got proteins that literally like sew them together, so it ends up being this seamless structure. There's also then these gap junctions that are very, very similar to plasmodesmata in order to be able to easily transport and transfer nutrients from cell to cell. Or we also have these things that are called these desmosomes that kind of just anchor, like almost like nail a cell to another cell in order to create some different types of functions. So really it's just knowing that multicellular organisms also need to have a way to exchange nutrients or adhere themselves to other cells. So you do have a nice summary at the back of your chapter of chapter 4, just talking about different sizes of cytoskeleton. So from a microtubule to a microfilament versus an intermediate filament, you don't have to know the difference between them. But if you hear microtubule, microfilament, or just filament, know that these are protein structures that make up the cytoskeleton internal structures is what they're for. You also have a nice summary of all of the different organelles and cell components that we're going to be finding, from the nucleus to your endoplasmic reticulum, to your ribosomes, to your rough ER, Golgi apparatus, lysosomes, vacuoles, mitochondria, chloroplast, and peroxisomes. So you definitely need to know those structures, which does take a little bit of memorization. But it really then becomes that application of what types of cells would have more ribosomes in it versus another cell. What type of cell would you expect to have more surface area compared to volume? So